Yes. Very good evening to all of you. Today I'm going to start with about cell cycle and cell division. We have done in the last very class we have completed up to prophase stage. So what we need to do right now is we are going to start from metaphase stage. But before I start from metaphase stage, I think we have to talk a little bit about whatever we have completed in the last very class. So the topic is about cell cycle and cell division. Now cell cycle and cell division. Normally we know that the cell division is performed by eukaryotic cells and some of the bacteria also. Now we know that the cell cycle, it is a sequential event. एक के बाद एक sequence में आते हैं. So they come in a sequential event. Number one. Number two is when we talk about the cycle timing. In case of human cell, जब human cell को culture किया गया in tissue culture, in culture device, we have found that for human cell to divide, it takes some 24 hours. कितना समय लिया? 24 hours. Now within this 24 hours, because we know that in cell cycle, there are mainly two phases. We can classify them broadly into two phases. One is M phase and the one is interphase. M means mitotic phase, a division phase, and the next one is interphase. What is interphase? Interphase, if you compare between interphase and M phase, which one is the largest one? The interphase is the largest one. So I am not holding right now. मैं बोर्ड में नहीं जा रहा हूँ इसको समझाने के लिए. I think you know this. I am just recapitulating some of the events. So in this whole cell cycle, the M phase is the slowest phase. सबसे बड़ा phase क्या है? That is interphase. And in the interphase we know that there are three phases. What are they? G1, S, G2. If you remember, we have talked about that in this interphase stage, the G1 takes place, the timing for the G1 is close to 8 to 12 hours. कितना समय लेता है G1 कंप्लीट करने के लिए? 8 से 12 घंटा. Then comes S phase, 6 to 8 hours. Then comes G2 phase, 3 to 4 hours. Fine. So 24 hours में ये है उसका division. So if the question comes, सबसे ज़्यादा समय किसका है? G1, then S, then G2, and the last is M phase. So M phase का timing सबसे कम होता है. So we are going to talk about M phase today, because in M phase we know that there are two types of phases. What is that? Number one क्या है? Karyokinesis, and number two is cytokinesis. So in karyokinesis we know that there are four phases. What are those? Prophase, metaphase, anaphase and telophase. ठीक है? We are not getting into that, but हमें NCERT के line पे जाना है. We are going to go in the line by line concept of the NCERT, and we are going to talk about those also. So M phase is having four phases: prophase, metaphase, anaphase and telophase. And after that what happens? Karyokinesis complete हो गया, and what happens after that is the last phase called cytokinesis. But when we talk about G1, S and G2, which was the most important one, जब एक cell cycle के अंदर वो आता है, the cell cycle contains 2N and 2C. What is 2N? N is the number of chromosome, and what is C? C is the amount of DNA. C is the amount of DNA. यहाँ तक clear है? C is the amount of DNA. But the moment from G1 it gets inside S. S मतलब synthesis phase. What is the meaning of S? S means synthesis phase. So in synthesis phase what happens? Replication of DNA occurs. Replication of DNA के लिए what are required? There are many of the enzymes which are required. बहुत सारे enzymes लगते हैं. And all those enzymes are basically proteinaceous in nature. So all these proteins are produced where? In the G1 phase. In the G1 phase. ठीक है? Now replication हुआ. The moment DNA replication हुआ, that means copying of DNA occurred. So that 2C is going to become what? 4C. And if you remember, I have given you three particular chromosomes in G1. I have shown you in G1, अगर आपको याद रहे, in G1, if I have shown you three types of chromosomes, one is like this, and is like this, and is like this, and inside, in between they have got, what is this? Centromere. सो ये अगर G1 में है, दिस इज द फर्स्ट क्रोमोसोम, दिस इज द सेकंड क्रोमोसोम, दिस इज द थर्ड क्रोमोसोम. G1 में अगर ये है, one chromosome is having one chromatid, but in G2 one, इसका क्या होगा? What is going to happen over here? In G2 what is going to happen? In G2 the same thing and ये होगा, पर इसके साथ क्या होगा? ये क्या होगा? और एक chromatid बढ़ जाएगा. There will be another chromatid and this chromatid we are going to call it as a sister chromatid. इसको क्या कहेंगे? Copy of this sister chromatid. ये sister chromatid है. So one centromere with two of the chromatid. One centromere, one chromosome with two of the chromatid. One chromosome with two of the chromatid. बोलो समझ में आया कि नहीं आया? So this is in case of G1 and G2. But when we talk about in G1, it was what? The primary condition was 2n 2c. एक सेल में कितना था? 2n number of chromosomes was there and 2c is the amount of DNA. But in case of G2, what is going to happen? 2n 4c. ये 4c कहाँ से आया? इन दोनों के बीच में what is the other phase? The other phase is what? The other phase is what? G1 S G2. S phase में क्या हो गया? 2n 4c. S में क्या हो गया? 2n 4c. So middle में कौन है? S phase है and that is why ये 4c जब G2 में आएगा, it is also going to be having the same amount, that is 2n and 4c, and that is why you can see in G2 it is 2n and 4c. Any issues with this? Clear? But after G2 it needs to get into what phase? It needs to get into which phase? Into M phase. M phase का पहला क्या है? Prophase, metaphase, anaphase, telophase. So what is the question in short? What the question is? G1 सबसे ज़्यादा time, then S, then G2 and last is M. So we are talking about M phase today. M phase में we have to remember how many phases are there? PMAT. How many phases are there? PMAT. What is PMAT? The first P is prophase, then metaphase, then anaphase, and the last one is telophase. Fine. So if we talk about all these four, सबसे important phase कौन सा है? Now, sir, important सभी phase है. Now, the most important phase for your exam वो है metaphase. Why? Because this metaphase, in metaphase, today we will start from metaphase only. Metaphase में क्या होता है? What happens in metaphase? What happens in metaphase? Metaphase में क्या होता है? Meta के पहले क्या था? Prophase. In prophase, in a nutshell, let me give you the idea about prophase first, then let's get into metaphase. Let me give you some idea about prophase first. So आपको पता चल जाएगा metaphase में क्या पढ़ने वाले हैं. What happens in prophase? Prophase में tell me what is the chromosome number? 2n. And what is the amount of DNA? क्या आ गया? 4c. So एक cell है. This is one of the cell. Say animal cell. This is an animal cell. और animal cell में there is a double membraned nuclear membrane. And what you can see, there are fine thread-like structure with a dense substance present over here. कौन बोलेगा what is this dense substance called? Nucleolus. Very good. And what is the function of nucleolus? Who's going to tell me? What is the function of nucleolus? What is the function of nucleolus? The function of nucleolus is to make rRNA, ribosomal RNA. क्या नाम है इसका? Ribosomal RNA. Ribosomal RNA का function क्या है? It helps in the making the structure of ribosome. It helps in the making of structure of ribosomal RNA. It makes in the structure of the ribosome. And what is ribosome? It is the site for protein synthesis. Protein बनाने का site का नाम क्या है? Ribosome है. So inside the nuclear membrane you can see a very clear conspicuous nuclear membrane present over here. They have got a fine thread called chromatin. Then there is a nucleolus present, and in the prophase what we can see, क्या बनाया था यहाँ पे? Centrosome, remember? And centrosome के चारों तरफ क्या था? Microtubules together. Centrosome plus microtubule together called aster, and aster plus spindle fiber. यहाँ पे दो थे, fine, and they are going to start moving towards the opposite pole. They will start moving towards the opposite pole. If you remember in prophase, we talked about 4 particular phases. 4 phase बहुत important था. What was that? The nuclear membrane starts disappearing. the nuclear membrane starts disappearing. This was number one. This was number one. Number two क्या था? Number two, the chromatin, they are going to start condensing. They are going to start condensing. Start condensing. To make what? Chromosome. To make chromosome. Chromatin thread starts condensing. Chromatin thread starts condensing to make chromosomes. Fine. NCERT में there is a term called intertwined. So they are going to start intertwining to become what? To become chromosomes. And then third, the nucleolus starts disappearing. The nucleolus starts disappearing. Starts disappearing. And the fourth one, what is that? The centrosomes, they are going to start moving to the opposite poles. Centrosomes starts moving towards opposite pole, towards opposite pole. ये 4 point important है for prophase stage. And let us see the same thing in what the NCERT has got to say. ये हम देखेंगे NCERT इसके बारे में क्या बोल रहे हैं. Let us find out in NCERT. Fine. देखो सेल साइकिल में फाइन what is required in cell cycle? What is required in cell cycle? थोड़ा देख लो यहाँ पे. What is required in cell cycle? Cell division is a very important process in all living organisms. During the division of cell, DNA replication and cell growth. Tell me, cell growth कहाँ पे होता है? Which stage? In which stage the cell grows? G1, S, G2. Cell grows in all the phases, but it grows to divide. So mainly in the interphase, the cell grows. Fine. All these processes, the cell division, DNA replication, and cell growth, hence have to take place in a coordinated way. क्या बोला था मैंने? Sequential event. I told you, replication of DNA. It's a sequential event. एक के बाद एक को होना है, and that is genetically taken care of. Genetic expression होना चाहिए. Genetic expression should be there. It is genetically involved. Fine. To correct division and formation of progeny cells containing the intact genome. जीन के अंदर जो रहेगा वो next cell में ही जाएगा. The same genome is going to go to the next genome. Durva, tell me. This is actually what the genome which goes to the next cell. Complete genome next cell में जाएगा, daughter cells में आएगा. That happens in mitosis or meiosis. So in mitosis, the entire genome gets into the next cell or daughter cells के अंदर parent वाला gene ही जाएगा in the next cell, called in case of mitosis. The sequence of events by which a cell duplicates its genome synthesizes the other constituents of the cell. सिर्फ जीनोम ही डुप्लीकेट नहीं होगा, सिर्फ जीन ही रेप्लिकेट नहीं होगा, सेल में अदर जो कंपोनेंट्स हैं. व्हाट आर द अदर कंपोनेंट्स? ऑल द सेलुलर कंपोनेंट्स. क्या क्या नाम है उसका? माइटोकॉन्ड्रिया, ER, एंडोप्लाज्मिक रेटिकुलम, गोल्गी बॉडी, दे शुड आल्सो रेप्लिकेट. माइंड यू वन थिंग, यहाँ पे इन केस ऑफ प्रोफेज एक पॉइंट हम ऐड कर सकते हैं. one point we can add. And what is that? 1 point we can add. यहाँ पे एक पॉइंट हम ऐड कर सकते हैं. बोलो, what is that? What happens to the ER? What happens to the Golgi body? Golgi body ये भी disappear करेगा नहीं करेगा? These two also disappears along with nucleolus. Fine. Okay. Now let's come back over here. Now the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells. So दो डॉटर सेल में डिवाइड किया, सेल के अंदर जो कंस्टिट्यूएंट्स हैं वो भी डिवाइड किए, एंड ऑल्सो द जीनोम ऑल्सो डिवाइडेड, and is termed as cell cycle. Although cell growth in terms of cytoplasmic increase, when a cell grows, if you compare between nucleus and cytoplasm, बोलो किसका ग्रोथ ज्यादा होता है? साइटोप्लाज्म का या न्यूक्लियस का? किसका ग्रोथ ज्यादा होता है? cytoplasm growth. So cytoplasm increase is needed. Is a continuous process DNA synthesis occurs only during one specific stage in the cell cycle. Tell me which stage? In which stage the DNA synthesis occurs? S phase. S phase. That is synthesis phase. The replicated chromosomes, DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Adrian, so what is most important? The cell cycle is under genetic control. Tell me, what is the example I have given you last day? A patient with gallbladder stone. A patient with gallbladder stone has come down to you when you become doctor. Now the condition is called cholelithiasis. The condition is called cholelithiasis. अब बोलो, ये है gallbladder stone का condition. So you need to remove the whole entire gallbladder along with the stones. आपको remove करना है. You have to cut this duct. The duct of the gallbladder is called cystic duct. क्या नाम है? The duct of gallbladder is called cystic duct. So you need to remove it. You need to remove the entire gallbladder along with the stones, and the medical condition for that is cholecystectomy. Tomy means removal. So cystic means cystic duct and chole means gallbladder. So you are removing the gallbladder along with what? Along with the duct. And what is the duct? Cystic duct. Now when you are performing this, you have to cut in the conventional method. आजकल तो वो रोबोटिक सर्जरी आ गया है. Now in a conventional method you are cutting off the whole entire portion. तो यहाँ पे 9 सेंटीमीटर का एक कट लग गया. तो 9 सेंटीमीटर आपने काट के गॉलब्लैडर को निकाल दिया आपने. You have completely removed the gallbladder along with the stones and along with the cystic duct. तो यहाँ 9 सेंटीमीटर जो आपने कट लगा दिया, यहाँ पे से 1 lakh cells are required. How many cells are required? 1 lakh cells are required to heal, to repair this particular cut off portion. How many cells are required? Say for example 1 lakh cells are required. क्या होता है? 1 लाख एक सेल बनेगा क्या? Is it going to make 1 lakh one cell? No. Who is going to decide how many times it need to divide? It's a genetically controlled. कितने बार डिवीजन के बाद 1 लाख सेल्स बनेंगे, उसके बाद it is going to stop the cell cycle. Otherwise क्या होगा वहां पे? What can happen? A tumor can take place over there. And what is tumor? Uncontrolled cell division. Uncontrolled cell division का best example कौन सा disease है? Cancer. What is the real cause of that cancer? Something goes wrong with the genetic control, and the cell divides and divides and divides, and the cell becomes immortal. The cell becomes immortal. We will talk about that in class 12. Cancer हम पढ़ेंगे, वहाँ पे we are going to talk about this. यहाँ तक clear है ना? यहाँ तक clear है? So it's a genetic control. It's genetic control. कितने सारे सेल लगेगा वहाँ पे आपको heal करने के लिए? It's completely genetically controlled. It is not going to have more than what is required. तो यहाँ पे क्या था? yeast cell में कल्चर में कितना मिनट लगता है उसको डिवाइड करने में? 90 minutes. But for human cell 24 hours. For human cell it takes 24 hours. For E. coli bacteria it takes 20 minutes to divide. Fine? Okay. This is M phase, G1, S, G2, prophase, metaphase, anaphase, telophase, and then cytokinesis. And we are not getting into, yes, some of the portions which are most required. I am going to get into those phase. M phase में क्या है? अच्छा, देखो यहाँ पे. It is significant to note that in the 24-hour average duration of a cell cycle of a human cell, cell division proper lasts for only about an hour. About an hour. Tell me, you. This 1 hour is for which cycle?
M phase.
Only M phase. For M phase, उसके पास कितना, उसको कंप्लीट करने के लिए कितना समय लगता है? To complete it takes only 1 hour. Is this clear? Any issues with this? Onesha, is this clear? So can you just tell me? G1 8 to 12 hours. S 6 to 8 hours. G2 3 to 4 hours. Last one M 1 hour. See, and we are studying out of that 1 hour. We have already studied last class. We have completed prophase stage. Today we have to start from the metaphase. M phase starts with the nuclear division, corresponding to the separation of daughter chromosomes. Karyokinesis and usually ends with what? लास्ट में क्या है? साइटोकाइनेसिस। लास्ट में क्या है? साइटोकाइनेसिस। सो इंटरफेस 95% ऑफ द ड्यूरेशन ऑफ द सेल साइकिल इज इंटरफेस। कितना परसेंट? 95%. एंड 5% इज व्हाट? एम फेज. In human cell, lastly we talked about this. In human cell, 24 hours is the time taken. Out of that, 95% is taken by interphase and 5% for M phase. 5% by M phase. So the interphase divided into 3 for the phase, gap 1 S and gap 2. Fine. Now NEET 2020 में क्वेश्चन आ गया था. During G1 phase, the cell is metabolically active. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. So cell size grows but it never replicates. DNA replicate नहीं होता है. DNA replication के लिए what is the phase? S phase is the phase. Okay. Now it came in 2020. Most of the organelles duplication occurs in G1 phase. Very very important. Organelle cellular component को भी duplicate करना पड़ेगा and that happens where? In G1 phase. Fine. Got the point? So this is it. S phase. S or synthesis phase marks the period during which DNA synthesis or replication takes place. Okay. How many years it came? 95, 96, 2001, 2010, 23. During this time, the amount of DNA per cell doubles. So amount of the DNA gets doubled. That means it was 2C initially. Now it has become 4C. What is C? C is called as the amount of DNA. She is called as the amount of DNA. Any issues? Now it came 2020. If the initial amount of DNA is denoted as 2C, initial amount क्या है? 2C. Then it increases to 4C. Increases to 4C. However, there is no increase. देखो इधर, there is no increase in chromosome number. Chromosome number remains 2n and the chromosome number becomes the amount of DNA becomes 2C. Initially क्या था? 2n 2C. Now it has become 2n 4C. That means chromosome number it never changes, but only the DNA amount gets doubled. Fine. In animal cells during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. In animal cell, देखो यहाँ पे very very important 2008 का क्वेश्चन है ये. In animal cells during the S phase, DNA replication begins in the nucleus. Replication takes place where? Inside the nucleus. And the centriole duplicates where? Inside the cytoplasm. Centriole का duplication कहाँ पे होता है? cytoplasm के ऊपर. When cell has stalled DNA replication fork, G2 or M checkpoint should be predominantly activated. अच्छा. When a cell has stalled DNA replication fork, G2 or M checkpoint should be predominantly activated. What is called checkpoint? देखो यहाँ पे checkpoint किसको कहते हैं? What is called checkpoint? What is called G2 checkpoint? Let us discuss about that. ये क्या है? This is G1, this is S, this is G2. This is G2. अब देखो ये बाद बाकी प्रोफेस কি নাম ছিল? বাবু কেটে দিয়েছিস? ওকে. প্রোফেজ, মেটাফেজ, অ্যানাফেজ, টেলোফেজ, PMAT. And what is this? This is karyokinesis. This is karyokinesis. What is this phase? This is cytokinesis. This is what? Cytokinesis. This is cytokinesis. This is cytokinesis. Fine. This is cytokinesis. Now what happens? A cell gets inside over here 2n 2c. 2n 2c. And when a cell gets inside over here, when it comes over in the S phase, क्या बन जाता है ये? 2n 4c. Now in this 2n 4c in cell cycle, during cell cycle, remember if this is DNA, double stranded DNA, अगर यहाँ पे A के साथ क्या है? What should be there? The DNA is 5' end and this is 3' end. This is 5' and this is 3' end. What is the base? Complementary base of A? T. और G के साथ C. G binds with C. But if there has been wrongfully, अगर T के अगेंस्ट में यहाँ पे A ना होकर गलती से T बना दिया देते हैं, fine. What has happened? There has been a mutation. T के अगेंस्ट में A ना होकर T बना दिया. So what has been formed now? It is called TT dimer. क्या कहते हैं इसको? TT dimer. And this event ये कहाँ पे होगा? This takes place when copying of DNA takes place. सोचो ये पेज को आप बोला गया आपको कि ये फोटोकॉपी करके लाओ, फाइन, तो फोटोकॉपी करके लाने के लिए बोला गया है. एंड इफ यू कीप योर हैंड लाइक दिस, योर पाम लाइक दिस, और इसका कॉपी बनाओगे तो हर एक पेज में क्या मिलेगा? इसका कॉपी मिलेगा ना. इन एवरी अदर पेज ओरिजिनल में अगर आपने ऐसे रख दिया तो इसका कॉपी मिलेगा. तो ये अगर है तो आपको पहले क्या देखना है? एक कॉपी करके देख लेना है क्या निकल रहा है. तो देयर शुड बी सम चेक पॉइंट ना, देयर शुड बी सम चेकिंग प्रोसीजर कि कॉपी थोड़ा देख लेते हैं. बिना कॉपी देखे आपने 100 कॉपी दे दिया, सो 100 कॉपी क्या मिलेगा आपको? ये हाथ रखकर आएगा ना वो. गॉट द पॉइंट? इज इट रिक्वायर्ड फॉर यू? नो. ये गलती कैसे हो गया? किसने अपना काम नहीं किया? चेक पॉइंट अपना काम नहीं किया. चेक पॉइंट. सो देयर शुड बी सम चेक पॉइंट. तो अगर टीटी डाइमर बन गया, इन रेप्लिकेशन देयर इज अ पर्टिकुलर एंजाइम व्हिच इज कॉल्ड डीएनए पॉलीमरेज 1. इसका काम क्या है? व्हाट इज द फंक्शन ऑफ दिस एंजाइम? इट हेल्प्स इन प्रूफ रीडिंग. And it reads whether there has been, if there has been some mutation. T के अगर इसमें T बन गया, is going to change T into what? Is going to change T into A. Is going to change T into A. Is this clear? So this is a checkpoint. Even अगर यहाँ पे चेक करना भूल गया DNA application missed to check it. So the next one is JIDU checkpoint. In the last class, I think I have talked about JIDU checkpoint. Yes, JIDU checkpoint. It is done by a gene called P53 gene. So ये इसको यहाँ पे इसको रोकेगा, सेल को यहाँ पे रोकेगा और चेक करेगा whether there has been some mutation occurred or not. If there has been कोई प्रॉब्लम हुआ कि रीडिंग करने में कोई प्रॉब्लम हुआ कि नहीं. फाइन. यहाँ पे सेकेंड टाइम चेक करेगा. फर्स्ट टाइम कौन चेक करेगा? इट्स a DNA application, DNA polymerase 1. Second time JIDU checkpoint, and it is done by a gene. I told you na, whole cell cycle is performed by genetic. It is under genetic control. So P53 gene का काम क्या है? The function of P53 gene is again to proofreading. अगर गलती से यहाँ पे TT आ जाता है तो वो क्या करेगा? Is going to change it into AT, and it has got another role. What is the role? The next role and most important role of P53 gene is apoptosis. This P is silent. And this is called as apoptosis. What is the other term for apoptosis? Is called as programmed cell death. क्या नाम है इसका? और एक नाम क्या है? Programmed cell death. That means every cell. जब सेल साइकिल में सेल कम्स ओवर हियर, JIDU checkpoint प्रोग्रामिंग कर देता है उस सेल को टू डाई. वो सेल के अंदर एक टाइम बॉम्ब इंप्लांट कर देता है. बिकॉज और वो टाइम बॉम्ब में कितना रहता है समय? सोचो 12 घंटा दे दिया वो, और वो टाइम बॉम्ब ऑन कर दिया, तो ये सेल इज गोइंग टू डिवाइड इनटू 2 सेल. 2 सेल में डिवाइड करेगा, पर हर एक सेल में क्या लगा हुआ है? कितना घंटा? 12 घंटा. 12 घंटे के अंदर दिस सेल इज गोइंग टू परफॉर्म इट्स फंक्शन. अगेन वो सेल, इस सेल के पास चॉइस है, 2 सेल, ये एक सेल घुसा था अंदर, अब ये दोनों क्या बन गया? आफ्टर साइटोकाइनेसिस कितना सेल बने? 2 ऑफ द सेल्स बीइंग फॉर्म्ड, एंड दीज 2 सेल्स आर डॉटर सेल्स. बट डॉटर सेल्स के अंदर टाइम बम है या नहीं है? अगर JIDU checkpoint सो रहा होगा तो टाइम बम रहेगा उसके अंदर? टाइम बम रहेगा? जॉल खा, टाइम बम रहेगा नहीं रहेगा? अगर टाइम बम नहीं रहेगा मतलब वो 2 डॉटर सेल्स अमर बन जाएंगे. दे आर नॉट प्रोग्राम टू डाई. एंड दे आर गोइंग टू बिकम कैंसरस सेल्स. बिकॉज उसमें क्या था? उसमें क्या रह सकता है? द म्यूटेटेड DNA कैन रिमेन ओवर देयर अनचेक्ड. गॉट द पॉइंट? अनचेक्ड. सो दिस इज द फंक्शन ऑफ what? JIDU checkpoint, and which is done by P53 gene, which is also called as apoptosis. And this can lead into what? क्या होगा वहां पे? ट्यूमर होगा नहीं होगा? अनकंट्रोल्ड सेल डिवीजन होगा नहीं होगा? क्योंकि चेक करने वाला कोई नहीं है. It's going to grow and grow and grow, and these cells are going to become immortal. These cells are going to become immortal. बोलो समझ में आएगा? यही बाद में जाकर क्या बनाएंगे? Cancerous cells. And what is the meaning of cancer? Cancer means uncontrolled cell division. Uncontrolled cell division. So यहाँ पे genetic control समझ में आ गया आपको. Fine. Now, prophase last day we have talked about what is metaphase. Let's get into that, and then we are going to come back to what NCERT has got to say. Now, metaphase. Metaphase में क्या होगा देखो. Already, what has happened? What has happened inside this cell? Inside this cell, this was what? What is this? This is centrosome. Centrosome. ये भी centrosome है. Now, so centrosomes have come down to the opposite pole. Centrosome have come down to the opposite pole. Fine. Centrosome plus microtubule is called aster. Aster से क्या बनेगा? Aster से क्या बनेगा? What is going to form in? Aster से क्या बनेगा? Spindle fiber. Very good. Now, this is one of the chromosome. This is one of the chromosome. Fine. This is one of the chromosome. और this is one of the chromosome. और ये उसका कॉपी है. क्या है? Remember, this is centromere. This is centromere. And यहाँ पे in centromere के पास there is a cup-shaped. There is a cup-shaped kinetochore. There is a cup-shaped kinetochore. Remember this? Kinetochore. याद आ रहा है? We have done this in cell chapter. Centromere contains a cup-shaped kinetochore. Kinetochore का function क्या है? What is the function of kinetochore? See, the chromosomes जो अभी आप देख रहे हो यहाँ पे chromosomes, these chromosomes they have arranged where? They have arranged themselves in the middle portion. अगर ये दोनों दो क्या है? Poles है. If these two have gone to the next pole, ये middle में this particular chromosome with how many chromatid? 2 chromatid. ये एक क्रोमैटिड है, ये और एक क्रोमैटिड है. बट कितने सारे काइनेटोकोर है? हाउ मेनी काइनेटोकोर आर प्रेजेंट? हाउ मेनी काइनेटोकोर आर प्रेजेंट? एक क्रोमोसोम के साथ कितने काइनेटोकोर है? 2. एंड दे आर प्रेजेंट विद द सेंट्रोमियर. वेरी गुड. नाउ यहां पे देखो दोनों क्रोमोसोम में सेंट्रोमियर आई हैव मेड इन द मिडिल. सो इफ यू रिमेंबर in the cell chapter, if the centromere is present in the middle, we call this chromosome as metacentric. We call this as metacentric. सोचो ये metacentric chromosome है with two of the cup-shaped structure called kinetochore. अब बोलो क्या बनेगा यहाँ पे? What is going to form in? See, this kinetochore is connected with the spindle fiber. The spindle fiber have been formed from here, from the centrosome, which is now called as पहले सेंट्रोसोम के साथ क्या था? माइक्रोट्यूब्यूल. एंड वी कॉल्ड इट एज एस्टर. One aster with two of the spindle, one aster with the two of the spindle makes it what? Mitotic apparatus or spindle apparatus. क्या कहते हैं उसको? Spindle apparatus. So हर एक क्रोमोसोम के साथ कितने सारे how many spindle fibers are attached with the kinetochore? 2. देखो ये एक स्पिंडल यहाँ पे और एक स्पिंडल यहाँ पे. So what it is going to do is ये देखो इसको ये एक है और ये एक ग्रीन वन. दिस इज द ग्रीन वन. एंड ये रहा यहां पे क्या है? काइनेटोकोर. यहां पे क्या है? काइनेटोकोर. एंड व्हाट इज पुलिंग इट? कौन पुल कर रहा है? स्पिंडल फाइबर. स्पिंडल फाइबर इज पुलिंग इट. सो स्पिंडल फाइबर्स आर ट्रेलिंग बिहाइंड. तो जब ये पुल करेगा इस तरफ, डू यू थिंक इट इज गोइंग टू रिमेन लाइक दिस? और ये ग्रीन वन, ये क्रोमैटिड किस तरह का शेप लेगा? ऐसा लेगा. दिस वन इज गोइंग टू टेक द शेप ऑफ दिस. दिस वन इज गोइंग टू टेक द शेप ऑफ दिस. और यहां पे कौन है? व्हाट इज प्रेजेंट? व्हाट इज प्रेजेंट ओवर हियर? फाइन. ये ऐसा नहीं रहेगा, ये उल्टा हो जाएगा ना. वी, ये वी हो जाएगा, ये उल्टा भी होगा. नाउ व्हाट इज पुलिंग बोथ ऑफ देम? ये कौन पुल कर रहा है? इस तरफ. ये पुल कर रहा है इस तरफ. व्हाट इज दिस? दिस इज स्पिंडल फाइबर. व्हाट इज इट? दिस इज स्पिंडल फाइबर. देखने में कैसा लग रहा है? वी लग रहा है. और यू टेल मी व्हाई इट इज लुकिंग लाइक वी? बिकॉज सेंट्रोमियर कहां पे था? मिडिल में था. सो मिडिल में रहेगा तो इट इज गोइंग टू शो व्हाट काइंड ऑफ शेप? वी शेपेड. और क्या जानते हैं? मेटासेंट्रिक के बाद क्या जानते हैं? व्हाट एल्स वी नो? दे कैन बी सब मेटासेंट्रिक. यहां पे है, ये छोटा है, ये बड़ा है. ये हो गया सब मेटासेंट्रिक. सब मेटासेंट्रिक मतलब क्या, व्हाट इज द शेप गोइंग टू बी? द शेप इज गोइंग टू बी एल शेपेड. वेरी गुड. एल होगा ना, इट्स गोइंग टू बी लाइक दिस, एल शेपेड, इज गोइंग टू बी एल शेपेड. और उसके बाद कौन है? ये सब मेटासेंट्रिक. उसके बाद क्या है? उसके बाद एक्रोसेंट्रिक, छोटा सा इधर है, एंड इट इज अ टेल एंड. टेल एंड में नहीं है यहाँ पे, तो ये इज गोइंग टू लुक लाइक व्हाट? जे शेपेड. जब ये पुल करेगा तो कैसा लगेगा? जे शेपेड. एंड द लास्ट वन इज टेलोसेंट्रिक. टेलोसेंट्रिक मींस एट द टेल एंड इट्स गोइंग टू लुक लाइक व्हाट? आई शेपेड. सो ये शेप किसके ऊपर डिपेंड करता है? द पोजीशन ऑफ सेंट्रोमियर in the chromosomes. Now the moment they are going to pull it, see यहाँ पे देखो, यहाँ पे क्या हो गया? यहाँ पे spindle fiber बना, यहाँ पे spindle fiber बना. So if the question is one chromosome is having how many kinetochore? 2. So in human body how many chromosomes are there? 46. So in metaphase how many kinetochore you can expect? 46 into 2. 46 into 2. Got the point? Clear? That is 92. Clear? So ये तो ऐसे these chromosomes they have arranged in which part of the cell? In the middle part or in the equatorial plate. So what is the plate called? Equatorial plate. What is the other term for equatorial plate? It is also called as metaphasic plate. क्या कहते हैं इसको? Metaphasic plate. Is this clear? So the chromosomes are going to arrange themselves in the metaphasic plate or the equatorial plate. Any problem with this? Come on. Fine. Any problem? कोई problem है? थोड़ा बुक में आप देखो in this diagram, इस पेज के last diagram पे, in the last diagram, you can see all the chromosomes are arranged in the equatorial plate. Clear? In the equatorial plate. So this is in metaphase. अच्छा यहाँ पे Varun, tell me, is there any nuclear membrane present? No nuclear membrane is present because nuclear membrane already disappeared in prophase. In prophase, the nuclear membrane completely disappeared. Nuclear membrane completely disappeared. So this is in case of metaphase. Tell me the chromosome number. How many chromosomes are there? Shonashish, how many chromosomes are there? 2N? 4C. 2N? The amount of DNA has remained doubled over here. And the chromosome number remained 2N. But what is the next phase? After metaphase, what is the next phase? It is anaphase. Anaphase. अच्छा, मेटाफेज. Why it is important? कोई भी सेल स्टेज को अगर आप माइक्रोस्कोप में स्टडी करना चाहते हो, सबसे एविडेंट फेज कौन सा होगा? नेम द फेज विच इज वेरी मच एविडेंट. मेटाफेजिक प्लेट. क्योंकि क्रोमोसोम्स दे आर गोइंग टू रिमेन कंडेंस्ड और दे आर गोइंग टू रिमेन अरेंज्ड इन द इक्वेटोरियल प्लेट इन द मिडिल पार्ट कॉल्ड मेटाफेजिक प्लेट. तो सबसे एविडेंट बेस्ट टू स्टडी फेज कौन सा है? व्हाट इज द बेस्ट टू स्टडी फेज? If you want to study, best to study phase है metaphase stage. Is this clear? Come down to anaphase. Come down to anaphase. Now in anaphase, anaphase. अच्छा, हिंदी में आना, किस तरफ आना? मेरे तरफ आना. कौन बुला रहा है इसको? कौन बुला रहा है इसको? This centrosome is calling it. The centrosome is calling it. यहाँ पे क्या है? Spindle fiber. क्या है ये? Spindle fiber. And then this is again chromosomes, sorry, centrosome with a spindle fiber right now. और वो किस तरफ बुला रहा है? आना. किस तरफ आना? पोल की तरफ आना. किस तरफ आना? पोल की तरफ आना. देखो इधर, this is one half, this is one half, V shaped, V shaped. Then this is another one, this is another one. All are V shaped because they are, all of them are, let's consider it to be metacentric. 1 2 3 4. Fine. ये 5. 1 2 3 4 5. बोलो समझ में आ रहा है कि नहीं आ रहा? So now in anaphase, this is for the first time they are going to become 2N 2C and 2N 2C. देखो, they are now splitting. This particular kinetochore, ये इसको pull कर रहा है towards the centrosome. Towards the centrosome, it is pulling it off. Clear? तो देखो यहाँ पे डिवाइड हो गया 2N 2C 2N 2C. Is this clear? Any issues with this? समझ में आ गया? So ये था एनाफेज में. एनाफेज इज द शॉर्टेस्ट फेज. अगर पूछा जाए ये चारों में से सबसे शॉर्टेस्ट फेज कौन है? एनाफेज इज द शॉर्टेस्ट फेज. एनाफेज इज द शॉर्टेस्ट फेज. Any issues with this? Okay. What happens in telophase? What happens in telophase? अब टेलोफेज में क्या होगा? देखो टेलोफेज में क्या होगा? टेलोफेज मतलब टेल एंड. टेलो मतलब टेल एंड, टेल एंड। तो ये कहां पे जाएगा? व्हाट इज गोइंग टू हैपन? सी इफ दिस इज अ सेल, इफ दिस इज अ सेल, व्हाट इज गोइंग टू हैपन? दिस इज अगेन सेंट्रोसोम, this is centrosome, okay, this is centrosome with the spindle fibers, now they have come down to the tail end. The chromosomes they have now arranged in the tail end. See, the chromosomes they have are chromatids. Now we cannot call it as a chromosome, now they have become chromatids. Also ये कहाँ पे चला गया? In the tail end. जब ये tail end में चला जाएगा, what is going to start forming in? The nuclear membrane starts reappearing. The nuclear membrane starts reappearing. देखो reappear करना start कर गया. It is just the opposite of what? Prophase. In prophase what happened? In prophase the nuclear membrane starts disappearing. और यहाँ पे क्या होगा? Nuclear membrane starts reappearing. देखो यहाँ पे भी nuclear membrane starts reappearing. So ये nuclear membrane starts reappearing. And what else? यहाँ पे तो chromosome था भी. These chromosomes they are going to start decondensing to form what? To form chromatin thread. पहले क्या हुआ था यहाँ पे? In prophase what happened with the chromatin? What happened with the chromatin? Chromatin ने क्या किया था? Condense करने के लिए वो start किया. They started intertwining and they have started condensing. They have started condensing. Decondensing to form chromatin threads. Chromatin threads. And what else? Nucleolus starts reappearing. Nucleolus starts reappearing. Third point. क्या सिर्फ nucleolus? Varun, tell me. Along with nucleolus, ER Golgi body. ER Golgi body. It also starts reappearing. It starts reappearing. Starts reappearing. Fine. And now the whole nuclear membrane have been formed. The whole nuclear membrane have been formed. The moment nuclear membrane completely gets formed, it is the end of what? क्या बन गया? दो nucleus division हो गया. It is called what? The end of karyokinesis. So what was there in karyokinesis? PMAT. Telophase complete हुआ मतलब karyokinesis complete हुआ. With the chromosome number what? 2N, 2C. 2N, 2C. So name the cell stage where the chromosome number again comes back to 2 and 2C. In M phase, it's anaphase. It's in anaphase. Any issues with this? Clear? So decondensation is a very very important point. Decondensation is very very important point. Fine. Let us find out. तो ये क्या हो गया? Karyokinesis हो गया. Now what is going to happen after karyokinesis? क्या होने वाला है? Cytokinesis. What is the next stage? Cytokinesis. Cyto मतलब cell. Kinesis मतलब movement. Cell का movement मतलब cytoplasm का movement. So what is going to happen right now? देखो इधर. This is a cell now, and the cell is having. This is the condition. This was the condition. Fine. Having the chromatin, having the chromatin threads, nucleolus. Fine. Chromatin threads. ये था condition. With the ER Golgi body, they are reappeared. अब क्या करना है? This is a cytoplasm. In case of animal cell, what is going to start occurring is over here. It's going to start showing off furrowing. It's going to start showing off furrowing. क्या दिखाएगा? Furrowing in animal cells. Furrowing is going to start taking place in animal cells. And आस्ते आस्ते these two are going to connect with each other. The moment they connect with each other, what is going to happen? There will be two cells. There will be two cells. There will be two cells. Fine. With 2N, 2C and 2N, 2C. When the cytokinesis completes in case of animal cell, you are going to have two daughter cells. You are going to have two daughter cells. Any issues with this? Come on, tell me. Any issues? कोई problem है बोलो? Is there any problem? Yes? Now, let us find out from NCERT. So what we have got? We have got two cells. अब बोलो ये cell, ये अभी डॉटर सेल है, डॉटर है, ये क्या पेरेंट हो गया? नो, इट नीड सम टाइम. इसके बाद इट हैज कम आउट ऑफ द सेल साइकिल. इट हैज कम आउट फ्रॉम द सेल साइकिल. इट हैज कम आउट फ्रॉम द सेल साइकिल. सो इट वाज G1, S, G2 and prophase, metaphase, anaphase, telophase, and this is cytokinesis. After cytokinesis it forms two cells. Now who is going to tell me what is the fate of these two cells? इसके अंदर टाइम बम है या नहीं है? नॉर्मली टाइम बम रहता है. एंड व्हाट इज द टाइम बम? द टाइम बम इज इम्प्लांटेड बाय P53 gene, which ultimately helps in apoptosis, which is called, it programs the cell to die. It programs the cell to die. Okay. अब ये the moment it has come out, daughter cells बन गया, it has got options. ये क्या हुआ अभी? It's going to grow in size. ये immediately क्या cell division में घुस जाएगा? It's going to come down in the cell cycle immediately. Or it can show some rest. It can come out of the G1 phase and get into quiescent phase. Sometimes it can show quiescent phase. What is that called? G0 phase. And what is G0 phase? It is not going to divide. ये permanently अभी dormant stage में जाएगा. It's not going to work. It's not going to be metabolically active immediately. It will take some time to become metabolically active. What about heart cells? अगर ये दोनों heart cells है, do you think heart cells divides? So if it never divides, एक बार डॉटर सेल बन गया तो वो और सेल साइकिल में नहीं जाएगा. Got the point? Heart cells, they show permanency. And what about neurons? न्यूरॉन डिवाइड करते हैं या सिर्फ रीजनरेट करते हैं? The number never increases, it only regenerates. बोलो समझ में आया कि नहीं आया? So this is what is in case of cytokinesis. This is what is in case of cytokinesis. Let's find out the year questions which came earlier. Okay, G2 pre-meiotic gap phase. In G2 see देखो यहाँ पे quiescent stage के बारे में कहा गया है. Yes. Now देखो इधर, during the G2 phase, the proteins are synthesized in the preparation for mitosis while cell growth continues. G2 में क्या होगा? प्रोटीन्स बनेंगे क्या करने के लिए? माइटोसिस करने के लिए. Now who is going to tell me G2 के बाद ये कहां पे आएगा? After G2 it is going to come down in M phase. In M phase, what is the protein which is required? कौन बोलेगा? Tubulin protein. Very good. That means it is going to make what kind of protein? It is going to make tubulin protein. And tubulin protein is going to be required for what? For the division. Some cells in the adult animals do not appear to exhibit divisions. Example? क्या लिया? Heart cell is never going to divide. It's there in your NCERT. And many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. अगर इंजरी हुआ कहीं पे, तो स्क्रैच हो गया, वहाँ पे 10,000 सेल चाहिए तो 10,000 सेल ही बनेंगे. एंड that is going to help in cell repair. Okay, these cells that do not divide further exit G1 phase. कहाँ से? Where from they are going to exit? G1. Where from they are going to exit? From G1. देखो, they are going to exit G1 phase to enter an inactive stage called quiescent or G0. That NEET 2023, it already came in the question paper. 23 NEET paper, you got this question. So it has to exit what? G1. ऐसा नहीं है कि immediately after division it is not going to get into G1 first and it is going to remain in G0. नहीं, G1 में जाने के बाद वो exit करेगा G1 के बाद. Is this clear? Any issues with this? Cells in G0 stage remain metabolically active but no longer proliferate. वो active रहेगा, वो dead नहीं रहता है. G0 में जो सेल जाएगा, it is not going to be dead. It is going to be metabolically active but no longer proliferate. उसका division नहीं होगा unless called on to do so depending on the requirement of the organism. बोलो समझ में आ रहा है क्या? If the organism never needs the cell to divide, it is not going to divide. So it is going to remain metabolically active. वो dead नहीं रहेगा, metabolically active रहेगा, but it's not going to proliferate or it's not going to divide. In animals, meiotic cell division is only seen in the diploid somatic cells. हम जानते हैं ना, last day we talked about. In animal cells, exception क्या है? In animal cells, it's going to take place where? In mainly diploid cell में mitosis होता है. only with the exception, कहाँ पे? In male drone, last day we talked about. In male drone, even they are haploid, because the haploid drones are mainly haploid. So we can see mainly in diploid cells, animal cells में division. But in case of plant cells, we can see the cell division in both haploid and diploid cell. In plant cell, we can see both in haploid and diploid cell. We have talked about that in the last very class. Against this, the plants can show meiotic division in both haploid and diploid cells. The correct sequence of phases of cell cycle is what? G1, S, G2, M. ये तो sequence हुआ. Even the time frame is also सबसे ज़्यादा टाइम G1, उससे कम S, उससे कम G2, उससे कम M. M में कितना 1, G2 में कितना 3 to 4 hours, 6 to 8 hours, और ये 8 to 12 hours, 8 to 12 hours. So important for your options. ये options में आएगा. Correct sequence of stages of cell division: gap 1 synthesis phase, gap 2 karyokinesis, and then last one is what? Cytokinesis. Okay. M phase or mitosis phase. Most dramatic period. Varun, tell me why it is called the most dramatic period. ज़्यादा ड्रामा होता है यहाँ पे. बहुत सारे ड्रामा होते हैं, बहुत सारे फंक्शंस होते हैं. मोस्ट ड्रामेटिक पीरियड ऑफ द सेल is M phase, involving a major reorganization of virtually all components of the cell. हर एक components में क्या होता है? Reorganization होता है. Every other cellular component they show what? They show reorganization. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. It is also called as, why it is called equational division? Because एक cell में, even after division, when it forms into two cells, it forms what? It makes the chromosome number remains the same. So ये chromosome number 2n था, after division the chromosome number remained the same, and that is why it is called equational division. So why it is called equational division? Because even after division, the chromosome number remains the same, and that is why it is called equational division. Okay. Though for inconvenience, mitosis has been divided into 4 stages of nuclear division. It is very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn between various stages. Mitosis is divided into the following 4 stages. What are the 4 stages? Prophase, metaphase, anaphase, and telophase. So there are 4 phases of mitosis. Let's talk about prophase and what we know, and just check it out. NCERT ने उनके बारे में क्या बोला है. Prophase is the longest phase. So what is prophase? Longest phase is prophase and the shortest phase is anaphase. Just now we have talked about. Longest phase is prophase and the shortest phase is anaphase. Prophase is the first stage of karyokinesis or mitosis. Follows the S and S-G2 of interphase. In the S and G2, the new DNA molecules formed are not distinct but intertwined. I use this term. I have used this term intertwined. But intertwined, very very important. Fine. DNA molecules formed are not distinct. Distinct नहीं है. They are intertwined. Even in prophase, what happens? Even in prophase, this is a stage. तो इनको क्या करना है? दोनों को अलग करना है. दोनों को अलग करना है. Intertwined. They are intertwined. Even after prophase, इसका function रहता है, and then they condense together. Remember last day we talked about euchromatin and heterochromatin. Which type of chromosome is going to be actively transcriptionable? Euchromatin are those chromatin which are not that condensed. Say for example, this is highly condensed. The chromosomes are highly condensed. तो इसका कॉपी बनेगा? टफ होगा. Highly condensed रहेगा तो इसको क्या कहेंगे हम? Heterochromatin. We are going to call it as heterochromatin. अगर उतना ज़्यादा कंडेंस नहीं होगा तो क्या कहेंगे उसको? Euchromatin. So transcriptionally active कौन सा होगा? Euchromatin is going to be transcriptionally active. What is the meaning of transcription? DNA से mRNA बनना और mRNA से DNA से mRNA बनने को क्या कहते हैं? Transcription. From mRNA to protein, what is that process called? Translation. What is that process called? Translation. Ankita, is this clear? Is this clear? Now, the chromosomal material becomes untangled. देखो क्या हो गया? Prophase is marked by the initiation of condensation of chromosomal material. So prophase is marked by, देखो option में ये रहेगा first, initiation of condensation of chromosomal material. So they are going to start condensing because in prophase what happens? The chromosomes remain in what form? Chromosomes are chromatin threads. So they start condensing to make what? Chromosomes. The chromosomal material becomes untangled during the process of chromatin condensation. So untangling होगा कब? जब chromosomal condensation होगा. The centriole, which had undergone duplication during S phase. So what is the phase when the centrosomes duplicates? It is during S phase of interphase. Now begins to move towards opposite poles of the cell. Now it has started moving towards the opposite poles of the cell. The completion of prophase can thus be marked by the following characteristics events. Very, very important. Number one, chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids. हर एक chromosome में कितना chromatid है? This is one chromatid. In case of this is in case of what? In case of G1. But now it is will be having two chromatids, a chromatid or a chromatid. Chromosomes seem to be composed of two chromatids attached together at the centromere. Where they are attached? What is the position? What is the location where they are going to attach? They are going to get attached in the centromere. देखो कहाँ पे वो attach करेंगे? दो chromatid, two chromatids, they are going to attach themselves where? In the centromere, and this will take place where? In prophase stage. Is this clear? Very good. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubule. हर एक centrosome से क्या निकलेगा? हर एक centrosome से क्या निकलेगा? This is one centrosome. This is one centriole and the centriole. Together, they are centriosome. And they have got microtubules coming out. They have got radiates microtubules called asters. The two asters together with spindle fiber. दो aster plus one spindle fiber. Very, very important. दो aster plus one spindle fiber is going to make spindle apparatus. Is going to make spindle fibers forms mitotic apparatus or spindle apparatus. क्या कहते हैं इसको? They are called as spindle apparatus or mitotic apparatus. This is very, very important. Mitotic apparatus. Is this clear? Any issues with this? Come on. Very, very important. Again, I repeat. Centrosome plus microtubule is equal to aster. One aster, two aster plus one spindle together makes spindle apparatus or mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes. क्योंकि क्या हो गया? Golgi complex has been disappeared. और उसके साथ कौन डिसअपियर हो गया? ER, nucleolus disappears, and the nuclear envelope, we have talked about that. So these are the stages in case of prophase. These are the stages which happens inside prophase stage. ये diagram में देखो, ये early prophase है. You can see the nuclear, the chromatin threads. You can see the dense particle called nucleolus. Now in the late prophase, what you can see? What you can see? Doubled. That means now what has happened? This has condensed. The chromatin threads, they have condensed to form chromosomes. This is in the late prophase, and the nuclear membrane disappears. This is the diagram of your NCERT. Fine. Prophase के बाद क्या? Prophase के बाद metaphase. And what happens in metaphase? You can see in the metaphase. You can see this is the equatorial plate. ये क्या है? This is the equatorial plate. These are spindle fibers and connected with what? बोलो? Kinetochore. Very good. Kinetochore. So एक क्रोमोसोम के पास कितने सारे kinetochore है? दो. So metaphase. This is equatorial plate or metaphasic plate. What comes then? Anaphase. What comes then? Anaphase. Anaphase में क्या हो गया? Pull हो गया. That means यहाँ तक क्या था? यहाँ तक क्या था? यहाँ तक था 2N4C. Very good. यहाँ तक था 2N4C. So 2N4C, यहाँ तक 2N4C. Prophase में भी 2N4C. But when it reaches anaphase, it for the first time it becomes what? 2N2C and 2N2C. So it moves towards the centrosome. Is in anaphase the shortest phase. Anaphase is the shortest phase. Any issues with this? Come on. Any issues with this? यहाँ तक clear है? What happens in telophase? बोलो telophase में क्या होगा? In animal cell, furrowing is going to occur. In animal cell, furrowing is going to occur and ultimately form into what? Telophase में क्या होगा ultimately? देखो telophase में क्या हो रहा है? What is going to happen? Nuclear membrane starts reappearing, nucleolus starts reappearing, ER, Golgi body starts reappearing, and what has happened? The chromosomes, they have started decondensing. They have started Clear है ना? And after telophase, what is going to happen? What is going to happen after telophase? What is going to happen? Again, they have formed into two daughter cells, and these two daughter cells are now, they have got choice now whether to go into quiescent or start proliferating. So first they are going to grow, and then they have a choice to get into what? Exit the G1 and get into what? Quiescent phase. Which cells can do that? Heart cells can do that. Heart cells, they are going to start permanency, showing permanency. Now in metaphase, the complete disintegration, देखो यहाँ पे term क्या है? Disintegration of the nuclear envelope. Nuclear envelope completely नष्ट हो गया. Nuclear membrane completely नष्ट हो गया. Marks the start of the second phase of mitosis. Hence the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed. So question, condensation of chromosome gets completed in which phase? It is in metaphase. And they can be observed clearly under the microscope, as because they get completely condensed, and that is why under microscope you can see them very, very clear. And that is the best stage to study is metaphase. Best stage to study is metaphase. This then is the stage at which the morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by centromere. We know this. Small disc shaped, क्या नाम है इसका? Disc shaped, Ankita, what is that? Kinetochore. Structures at the surface of centromeres. What is that? Kinetochores. These structures serve as the sites of attachment of spindle fibers formed by the centrioles to the chromosomes that are moved into the position at the center of the cell. बोलो ये तो हमने, we have completed that just now. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator. ये कहाँ पे arrange करेंगे? खुद को equator में. With one chromatid. है ना? टूट गया ना? Metaphase में. Now with one chromatid attached with one kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. बाबा ये क्या है? ये क्या है? If you remember? Yes, if you remember? If you remember, this is one. Fine. This is one. What is present over here? Kinetochore. And what is this? Spindle fiber. So that is what is being said. Fine. The plane of the alignment of the chromosomes at metaphase is referred to as the metaphasic plate. ये क्या कौन सा plate है? Middle वाला plate, metaphase plate. The key features of metaphase are. What are the key features? जहाँ से question आप expect कर सकते हो in NEET. What is that? देखो कितने साल आ गया? 2011, 2020. Spindle fibers attached to kinetochores of chromosomes. Chromosomes are moved to spindle equator and get aligned where? Along the metaphasic plate through spindle fibers to both the poles. Both the poles. Come down to anaphase. सबसे shortest phase anaphase में है. Fine. सबसे shortest phase है. Shortest phase में देखो. What is very important? Centromere split and chromatids separate. Centromere split and chromatids separate. Chromatids move to the opposite poles. Anaphase promoting complex, very very important, APC. ये शायद आपके न्यू बुक में नहीं है. APC has been removed. What is it? Anaphase promoting complex, APC, is a protein degradation machinery. ये क्या करता है? It feeds on the protein. It's a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in a human cell, then chromosomes will not segregate. अगर APC defective है तो chromosomes क्या नहीं होगा? Will not segregate. Segregation of chromosomes is not going to occur. So this is APC. This is very very important, but I think new book में ये off कर दिया गया है. Fine. This is anaphase, telophase, interphase. We have seen that. Now let us talk about telophase. Telophase में क्या है? Chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear membrane develops around the chromosome. देखो nuclear membrane आ गया. Nuclear membrane reappears. Nucleolus, Golgi complex and ER reform. Last point. Cytogenesis. What is cytogenesis? We have seen. In animal cell we have seen the cytogenesis कैसे होता है. But what in case of plant cell? Just a moment. In case of plant cell, what happens in case of plant cell? In plant cell, this is the cell wall. This is the cell wall of a plant cell. Cell wall of plant cell. And in the middle, for the first time, a calcium pectate is going to start forming in. And this secretion is going to come from the Golgi body. This secretion will come from the Golgi body. Fine. And this calcium pectate is going to be in the middle. It's going to start from inward to outward. And already what is present over here? Two of the nucleus. हम क्या देख रहे हैं? Cytokinesis in plant cell. Cytokinesis in plant cell. We are watching cytokinesis in plant cell. कैसे cytokinesis होता है in plant cell? Furrowing नहीं होता है. It never furrows because cell wall is rigid. Cell wall is a rigid structure. So calcium pectate is going to start forming in, and this calcium pectate, which is a secretion of the Golgi body, starts moving from the inward to outward and ultimately forms total. It makes like this. And what is going to happen? See, this is the cell membrane. This is the cell membrane and this is the cell membrane. And what is going to form in between? Cell wall is going to form in between and form what? Between two cells, what is this one? This is the middle lamella. Calcium pectate and magnesium pectate becomes the middle lamella. And this is being formed from the modified Golgi body. And what is the other term for this? Cell plate formation is also called as phragmoplast. Cell plate formation is also called as phragmoplast, cell plate formation. Fine. So this is the way that cytokinesis occurs in plant cells. You may go now. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell. कहाँ से? From the center of the cell, and grows outward to meet the existing lateral walls, center to walls. देखो, अंदर से बाहर की तरफ, inward to outward, and they form what? They are going to connect the wall. The formation of this new wall, cell wall, begins with the formation of a simple precursor called the cell plate. Simple precursor. Starting, कौन करता है start ये? Cell plate. It came in 2004. Cell plate is called as, come on, phragmoplast. Is this clear? Which makes the cell plate. That represents the middle lamella between the walls of the two adjacent cells. Ultimately, this makes what? This makes the middle lamella. This is all about the plant cell and animal cell, how they divide. Fine. Sometimes what happens? अगर सोचो, there is a particular component. There is a particular component called colchicine. क्या नाम है? Colchicine. क्या कहते हैं इसको? Colchicine. It is called as a mitotic poison. क्या कहते हैं इसको? Mitotic poison. क्या करेगा ये? It is going to feed on the spindle fiber. It feeds on spindle fiber. It feeds on spindle fibers. Think about here anaphase है. ONESHOT, tell me. If this is anaphase, and what is required in anaphase? This is going to happen. Fine. These chromosomes are going to start moving towards the opposite pole. This is what is going to happen. V-shaped chromosomes are present. Now, what is the stage? 2N, 2C. 2N, 2C. And now the spindle fiber, if this anaphase stage is having colchicine, it is going to feed on what? Spindle fiber. If it feeds on the spindle fiber, it is going to result in polyploidy. What is polyploidy? Many chromosomes are going to remain in one cell. It's going to remain in one cell, and we call it what? Multinucleated conditions. Remember, in fungus, we have talked about synocytic or syncytium. What is the term called? Syncytium. Multinucleate condition. देखो, at the time of cytoplasmic divisions, organisms like mitochondria and plastids gets distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate conditions arises, leading to the formation of syncytium, that is liquid endosperm in a coconut. Acha, colchicine is not being given in your book. Okay? What it feeds on? Colchicine एक माइटोटिक पॉइजन है, वो किसके ऊपर फीड कर लेता है? स्पिंडल फाइबर के अंदर. So what is going to happen? The chromosome number is going to increase, called polyploidy, and this we can call it as, now what is called syncytium? Multinucleate conditions. वो एक ही सेल में बहुत सारे न्यूक्लियस प्रेजेंट रहना, और वो कहाँ पे रहता है? In the coconut water, the liquid endosperm that we feed in, they are having many of the nucleus They have got, in one cell, they have got many nucleus. Because what is not going to take place over there? Karyokinesis is going to occur. Nuclear division होगा, but not followed by cytokinesis. अगर cytokinesis हो जाता तो क्या होता? दो सेल बन जाता. But एक ही सेल में दो nucleus. This condition, or दो से ज़्यादा nucleus. This condition you can only see if there is no cytokinesis. If the cytokinesis is not going to take place after karyokinesis. What is the significance of mitosis? Mitosis का significance क्या है? Without mitosis, you cannot have any, repair of cells. Repair of cells. अगर जहाँ पे cell damage होगा वहाँ पे repair होना चाहिए. So what is, and also the chromosome number remains the same. So देखो यहाँ पे mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, क्या नाम क्या है? B. Haploid cells also divide by mitosis. Mitosis division. Mitosis results in the production of diploid daughter cells. देखो, mitosis से क्या होगा? Diploid daughter cells बनेंगे. Mitosis is going to help in. It results in the production of diploid daughter cells with identical genetic complement. Usually, the growth of multicellular organisms is due to the mitosis. Multicellular organisms cannot grow. किसका mitosis ज़्यादा होगा? डायनासोर में या हेमीडैक्टाइलस में, लिजार्ड में, हाउस लिजार्ड में? इन डायनासोर ओनली. गॉट द पॉइंट? उससे क्या होगा? सेल नंबर किसमें ज़्यादा है? डायनासोर में. तो वहाँ पे mitosis ज़्यादा हुआ है. Results in disturbing the cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleus-cytoplasmic ratio. अच्छा देखो यहाँ पे बहुत important point है. ये एक cell है with a nucleus. The moment the cell grows, nucleus size तो ज़्यादा नहीं बढ़ेगा. कौन बढ़ेगा? cytoplasm का size. तो cytoplasm का size अगर nucleus से cytoplasm size बढ़ गया, it has to divide immediately because cytoplasm का size बढ़ गया. बोलो समझ में आया कि नहीं आया? बोलो समझ में आ रहा है? So nucleus-cytoplasm ratio maintain करने के लिए what is required? Mitosis is required because this is the nucleus and this is the cytoplasm. See the amount is, say for example, the amount is same to same. Same to same नहीं रहता है. So यहाँ पे 2 है तो यहाँ पे 4 होगा. पर यहाँ पे क्या हो गया? यहाँ पे 2 ही है पर ये 8 बन गया. तो यहाँ पे क्या होगा? 2 और 4 और 2 और 4. बोलो समझ में क्या नहीं आया? So to maintain the same particular nucleus-cytoplasmic ratio, the cell needs to divide. A very significant contribution of mitosis is cell repair. A very significant contribution of mitosis is cell repair. These are the 3 points. The cells of the upper layer of the epidermis, skin में, cells of the lining of gut के lining में, and blood cells are being constantly replaced. Blood cells are being constantly replaced. Now tell me, यहाँ पे एक twist है. And the twist is in blood cells. Blood cells के बारे में बोलो. RBC, WBC, platelet. ये divide करते हैं नहीं करते हैं? Do they divide or they are formed inside bone marrow? They are formed inside the bone marrow and that's why they are called formed elements. They are called as formed elements. तो सबसे ज़्यादा repair कहाँ पे चाहिए आपको? gut में चाहिए. सबसे ज़्यादा repair कहाँ पे चाहिए? Skin पे चाहिए. Every day what happens? The upper layer. If you consider the skin के एकदम top layer में क्या रहता है? Stratified epithelium. That means it is having stratum corneum, the upper layer which becomes dead due to the harsh environment. और उसके बाद from below what is there? Stratum germinativum, which is also called as stratum malpighi. वहाँ से क्या बनता है? New cells बनते हैं. They form into new cells and they heal up those dead cells. Fine. So that is very very important. So सबसे ज़्यादा कहाँ पे होता है? Gut cells में. Now we have solved some of the questions except some. Last day we have solved some of the questions. Let us find out. What is this? Start with this. Some dividing cells exit the cell cycle. Some dividing cells exit the cell cycle and enter vegetative inactive stage. This is called quiescent stage. This process occurs at the end of Option A, G1 phase. End of G1 phase. Next, identify the correct statement with regard to G1 phase of interphase. G1 phase of interphase. Correct statement. And give it a try. Reorganization of all cell components takes place. Cell is metabolically active, grows but does not replicate.
Sir, option B.
Option B. What is that? Cell is metabolically active, grows.
But does not.
Identify the correct statement with regard to G1 phase. In G1 cell is metabolically active, grows but does not replicate its DNA. Fine, very good. Well, in a meiotic cell, the correct sequence of phases is very easy. G1, S, G2, M. Cells in G0 phase exit the cell cycle, enter the cell cycle, suspend the cell cycle, terminate the cell cycle. Exit the cell cycle. G0 de ki hoy. What happens in G0? What happens in G0? The cell exits the cell cycle and they remain in a permanent stage. They are not going to divide. They are not ready to divide. Okay. Yes, very good question. It's a very good question. Tell me. Given below is a schematic breakup of the phases. You can see A, B, C. Mitosis mein. What is A? These four stages together makes what? Karyokinesis. So A is karyokinesis. Which one of the following is the correct indication of stage phase in the cell cycle? A is cytokinesis. A is karyokinesis. Ki ho gaya? Which one of the following is the correct indication of the stage or phase in the cell cycle? C is what? C is cytokinesis. D, synthetic phase. Okay, this is G1, this is synthetic phase, D synthetic phase, okay. S, where is G2 then? This is interphase, this is M phase. Okay, this block, this block, this block and this block. Oh God, this block is not been defined. What is it? Which one of the following is the correct indication of the stage phase in the cell cycle? A, B, C.
Option B, sir.
Option D. B is metaphase. B is metaphase. Where is B? This prophase metaphase. No, B is not metaphase.
Option B, sir.
Option B is D synthetic phase. D synthesis phase is right. Because we can consider this as G1, S, G2. And then this is M phase. Okay? At what stage of cell cycle are histone protein synthesized? We have done it already. Histone protein बनता है कहाँ पे? कहाँ लगेगा histone protein? Where it is needed?
For formation of spin.
No, histone protein, the DNA wraps around histone protein to condense it. Now bolo. At what stage of the cell cycle are histone protein synthesized in a eukaryotic cell? During G2 phase, G2 stage of prophase, during S phase, during entire prophase, during telophase. During G2 stage of prophase, histone. Remember, we have talked about G2 में क्या होता है? G2 में 2N4C बन गया कहाँ पे? In S phase. In S phase, 2N4C have been formed, but for the DNA, ये जो DNA बन गया, S phase में DNA बन गया. In S phase, DNA has been formed. Now this DNA needs to wrap around the histone protein. This DNA needs to wrap around the histone protein like this. Come on. This is a histone protein, and wrapping around the histone protein with the H1 protein, we call it as nucleosome model. क्या कहते हैं इसको? Nucleosome model. तो histone protein बनेगा कहाँ पे? During G2 stage of prophase, during S phase में बनेगा? No. During entire prophase तो बाद में है M phase में. During telophase वो भी तो M phase में है नहीं. So what is that? During G2 stage of the prophase. Yes. In the somatic cell cycle, करो ये. In the somatic cell cycle, in G1 phase, the DNA content is double the amount of DNA present in the original cell. DNA replication takes place in the S phase. A short interphase is followed by a long meiotic phase. G2 phase follows meiotic phase. Tell me.
Sir, option B.
In somatic cell, what is somatic cell? The normal cells. We have got two types of cells. One is somatic cell and another one is germ cell. In somatic cells, that is diploid cells, DNA replication takes place in S phase. Answer is B. In G2 phase follows. Achha, in cell cycle, DNA replication takes place in again S phase. S phase. ये question आप comment करो मुझे. This question is very, very important. A bacterium divides every 35 minutes. If culture containing 10 to the power 5 cells per milliliter is grown for 175 minutes, what will be the cell concentration per ml after 175 minutes? Who is going to do it? Or you need time to do it? 175 मिनट्स कितना टाइम्स है? 35 का कितना टाइम्स है? हाउ मेनी टाइम्स? हाउ मेनी टाइम्स? कम ऑन, 5 टाइम्स. 5 टाइम्स. तो 5 टाइम्स डिवीजन हो गया. And what is the normal cells? You are having a culture containing 10 to the power 5 cells per ml. तो कितना होगा? Already initial concentration कितना है? Initial numbers कितने हैं? 10 to the power 5 number of cells were there in the initial concentration. Initially. And how many times? 35 minutes में बैक्टीरिया डिवाइड करते हैं. Every 35 minutes में बैक्टीरिया डिवाइड करते हैं. 175 मिनट के बाद बैक्टीरिया कितना टाइम्स डिवाइड करेगा? 5 टाइम्स. तो 5 टाइम्स डिवाइड करेगा. अगर 10 to the power 5 है इनिशियली तो क्या होगा वो? कम ऑन, 10 to the power 5 होगा, 10 to the power रहेगा. बट सी द ऑप्शंस, हाउ मेनी टाइम्स इट हैज बीन डिवाइडेड. कौन सा लेना है? 2 to the power n. And what is the n over here? 5 times. So 2 to the power 5 is what? 2 to the power 5 is what? 32. Take time and do it. Take time and do it. This is the answer. Which one of the following structures will not be common to biotic cells of the higher plants? Not common. No. Which one of the following structures will not be common to biotic cells of higher plants? Plants में centrioles नहीं रहते हैं. Plants में centriole नहीं रहते हैं. Cell plate बनेगा, centromere रहेगा, spindle fiber रहेगा. But what is not there in plant cells? Centriole is not there. That's all about the chapter, not chapter. That is all about the mitosis phase. So next day what we are going to do is about meiosis. Go line by line of the NCERT. Start solving the questions. I'm going to send this in your in Telegram channel when I'm going to send it to you. Fine, so that you can solve some questions. Okay, Babu. So next day क्या करेंगे हम? We are going to start with meiosis. The most important. Just एक बार देख के आना and we are going to complete that. Okay? चलो, all the best.
Thank you.